I'm presenting on genetic biocontrol in common carp, as well as directed evolution of the enzyme CRISPR-Cas9 for low temperature catalysis. And I'm mostly focusing on the first half of that title. I'll mention the second half. I'm putting a lot of work into that, but for the sake of this presentation, I'm going to mostly just skim over it. So invasive species as a whole have a really significant cost, environmentally as well as economically. It's estimated that in the US alone, that cost is somewhere around $120 billion, with the breakdown of where those costs are coming from shown in the pie chart on the right. So it's kind of, there isn't any like one particular group that's causing all of that cost. With invasive species, there's a lot of focus on prevention of just them getting into somewhere in the first place, because the cost as they start to establish themselves in a location increases significantly, to the point where once they're very well established in an environment, it's really hard to get rid of them, and usually we don't do that. Genetic biocontrol has some advantages over a lot of the current methods of getting rid of invasive species or pest organisms, because it does like pesticides, and it can be tailored to the species that you're trying to target. So whereas like a fish toxin might get rid of the common carp, for example, it's going to also target a lot of other species. Genetic biocontrol works through reproduction though, so it only would work on an organism that can reproduce with the one that you're targeting, making off-target effects a lot more difficult. And because genetic biocontrol can be made with a range of different persistence or aggressiveness, it's very scalable. You can make it so that a very small release of a few genetic biocontrol agents could wipe out a large population, or you could design it in a different way so that you need to keep releasing it over time in order to reach the desired effect, making it so that the method you use can be tailored to the area you're trying to treat. The idea would be that we would combine this with existing methods. For example, with the carp, net out a whole bunch of carp first. Those are those physical and chemical methods to knock down the population, and then throwing in the biocontrol agent carp in order to further reduce and possibly even eradicate a population through that unproductive mating. One of the methods of genetic biocontrol that I'm focusing a lot on in my research are gene drives. And gene drives work by using a Cas9 enzyme, which binds to a guide RNA, and the combination of that Cas9 enzyme and guide RNA allow you to specify a very specific sequence in the DNA of, in this case, the carp, that you want the enzyme to cut. So in the schematic on the right, you can see that there's the Cas9 guide RNA targeting a really specific site. And once you create a cut in the DNA, the organism can either repair it by homology-directed repair, which is very precise. It copies from another chromosome and takes that exact copy and puts it onto the spot that was damaged. It can also use non-homologous end joining, where it just kind of sticks them together. That can result in mutations, extra DNA and stuff. And these are going to be a little bit more important in understanding how the gene drives work. Gene drives are one of the more aggressive forms of genetic biocontrol, but even within that, they are tunable. So the way that works is they have higher than Mendelian inheritance. The gene drive will cause itself to be copied onto a gene drive parent will only have gene drive offspring, I think is how I'm going to explain it for this slide. So with Mendelian inheritance, you'll see here that like 50% of the offspring will receive this blue gene here. So it gets lost in the population over time. But with gene drives, every offspring produced by a gene drive insect will be a gene drive insect itself. So over time, the population gets replaced with these gene drive organisms. You can vary the levels of aggressiveness so that you can tune it to a specific population. And these are models of gene drives using data on how they performed in the lab, showing that you're converting the population over to a gene drive genotype, and you can also use them to knock out a population. So in the right figure, you have the fitness of a population, and you see as the gene drive allele gets higher in the population, the fitness of the population as a whole decreases. Another form of genetic biocontrol that my lab is working on is sex ratio biasing methods. And the way that these work is you have, for example, female specific is usually the one that's most helpful in fish. So you have a promoter that's specific to females, and it's driving some gene. Usually you want that to be something lethal in the organism. And when you do this, you'll end up with males that can survive and pass on that gene, but females that cannot. This has been tested in zebrafish as well as the olive fly in lab conditions. And with the female lethal genes, you see that the population really dips after the release of males carrying this female lethal gene. This was also modeled to be combined gene drive and female lethal at the same time, as shown in the left. And then on the right, there were cage studies performed with gene drives that were combined with female lethal. And these make it act more aggressively because the combination of the two makes it so that females can't survive, but males pass on 100% of that gene to every fly, in this case, that they mate with. As for the common carp are destructive when they get into aquatic ecosystems. They like to root around in the bottom of lakes and rivers, and in doing so, they pull out vegetation and stir up a lot of sediment. That sediment turns into algae, and you get eutrophication of the body of water. The sunlight can't reach the bottom layers as well, and you'll get further plant death. And over time, they alter the entire ecosystem to the point where, where they're the only ones that are really able to survive there. They're very widespread. The common carp have gotten all over the place. In the US, we brought them here as a food source. They're also a really big problem in Australia. The current management strategies for the common carp are really expensive and really labor intensive, and they can't get rid of the carp entirely, but rather just maintain a population that is less destructive. One of the most common strategies, other than just tracking them and preventing them from getting further, is removing them with nets. And that has to be done over and over again. As you can imagine, any carp that escape the netting will just go and reproduce, and over time, the population builds up rather quickly, so then you're netting year after year after year. Other strategies that are being used or proposed are chemical treatment. These are general fish poisons usually, so they aren't specific to the common carp, but the idea is you would wipe out the carp, taking out some other fish while you're at it, and then restock with the fish that have been killed. This is not ideal, but it can reduce or remove carp. Biological control methods, I mean, predation is not sufficient for the common carp. They can be eaten when they're very small, but since they get so big, there aren't really predators here that like to eat them as fast as they can reproduce. And the koi herpes virus is being proposed as a solution in Australia. However, the koi herpes virus could spread to koi, which we aquaculture a lot here, as well as to carp that are in farms where we don't want them to be dying, getting sick, etc. There are some challenges to developing genetic biocontrol in the common carp specifically, that also make it an interesting test of how well genetic biocontrol performs outside of insects, in a lab environment. So they're a non-model organism, and to date, all of the genetic biocontrol tests, specifically gene drives that I've looked into, have been performed in insects that are model organisms. So you have the challenges with a longer maturation time and just there not being protocols for working with this organism, especially doing transgenesis. They also have a recent genome duplication. So some of their genes are duplicate, and there are extra copies of them. Others have had enough time to diverge. So there's a lot of questions about, like, if you're choosing a gene to target for something, is that gene going to, like, are you targeting one copy or two copies? And can you, are the sequences similar enough for you to do that? The third challenge that we're facing is that the molecular tools that have been utilized for genetic biocontrols are designed to function in lab environments, and some of them don't work as well in the cooler environments that the aquatic, like common carp, prefer to be in. So the main goals based on those three challenges are: one, developing protocols in year-round spawning and transgenesis. So protocols for that, for the common carp specifically, so that we can have access to embryos, and creating transgenic fish year-round. Aim 2 is designing the genetic biocontrol elements with that genome duplication in mind, specifically focusing on how will that genome duplication affect, like, the homing of a gene drive. And aim 3 is development of cold-tolerant CRISPR-Cas9, which I'll touch on, but I'm not going to dig into too deep. So for aim 1, in the wild, the common carp like to spawn annually, once, maybe twice. And if we were to do transgenesis using carp spawning in the wild, you'd imagine we'd have to get on a boat, we'd have to go out there, catch them just when they're spawning, scoop them up in a net, and collect those eggs, hurry them back to the lab. And if we were to attempt transgenesis that way and it didn't work, we'd have to wait a whole nother year and try again. So that's just not going to help us advance this technology very quickly. So instead, we took carp into the lab and tried to get them to spawn in lab conditions. To do this, we took the carp and we gave them all pit tags so that we can keep track of who's who. And we rotate them in small batches through artificial temperature and daylight cycles in order to trick them into thinking it's a season that it isn't. So after a spawn, we give them some time in a summer phase to develop eggs, and then females with a very large developed tummy are brought down into a winter phase so that those eggs can arrest, and it signals to the carp to, like, let those eggs mature. We then bring them into a spring phase for the final maturation of the eggs before giving them a quick 4-day increase in temperature to let them know it's time to spawn, and then we induce spawn. We've been able to get access to embryos about once every 2 weeks this way. Again, there's like sometimes it doesn't work. We're working with biology of a fish, so they can be stubborn sometimes. And then we developed protocols for transgenesis. To do this, we used GFP. It's a gene that causes a fish to glow. I guess fluoresce, to be technical here. So the reason that we use this is we could tell if the protocol was working right away because the fish is green. You can just kind of see it. And then we could use that readout to optimize how well our protocol was working. We did successfully create transgenic fish using protocols, and we've now optimized and have started introducing genetic components for genetic biocontrol methods instead. But we let one batch of the GFP fish mature to show that it got into their germline. You see that these fish are kind of speckly. Well, I guess this fish, his name is Phineas. He's kind of speckly because the gene didn't make it into his genome like right away when he was in the 1-cell stage. So some of his early on development cells got that gene and others didn't. And the result is a very speckly fish. And that speckliness, that mosaicism, is also present in his germline. But we were able to confirm that 15% of his offspring are also transgenic. So we can make it into the germline, we can pass it on. The next goal was to actually make the genetic biocontrol elements. Now that we can do transgenesis in the common carp, let's do something with it. So carp have a genome duplication event. Some terms here that are kind of important are paralog. Those are homologous genes resulting just from a genetic duplication somewhere. So in this diagram here, can you guys see my mouse? Anyway, so those are going to just be like a gene copies itself somewhere else. Whereas an onolog is a paralog that results from the duplication of the entire genome. So most teleosts already have 2 copies. The common carp has 2 copies of a lot of genes, but it can also have extra copies, or called onologs, from that recent genome duplication. And it was long enough ago that some of them have mutated and become non-functional or gained other roles in the fish, while others still retain their original function. Duplicate genes can evolve divergently, so you can get loss or retention of the original function. Key components of the gene drives that I'm working on are the I have them pulled out here individually. So on the end here we have GFP. That's the same reporter that is being used to make the fish glow green. This will just help us to see if that gene drive is being passed along at a glance. We've also got a promoter that is only active in the germline of the fish. And this is important because we're doing a suppression gene drive. Basically, if a fish has 1 copy of the gene, it can still survive. If it has 2 copies of the gene drive, the idea is that it won't survive. And by making the Cas9 only express in its germline, we make it so that this gene drive here, as you can see in the little schematic, the Cas9 will be expressed in the germline. It will cut the native DNA, which will get copied of the gene drive, and you'll result in gene drive only genotype, but only in the germline, so that the fish can still survive with its 1 copy of the wild type DNA. But it can only pass on gene drive DNA. The 2 promoters specifically that I picked out were Peewee, which is called Ziwi in the zebrafish, and Vasa, which both of them are only expressed in germ cell development, which is ideal for this type of gene drive. Yes, and then I've got guide RNAs expressed by U6 promoters, so those will just kind of always be available. And the idea then is that those guide RNAs will cause the Cas9 to cut at the beginning and the end of an essential gene. So the genes that we're picking to target are CTCF, which is essential and haplosufficient in zebrafish. It's important for chromatin structure. mTOR, which is also essential in zebrafish. We're hoping that one is haplosufficient, but we haven't found data on that. And ADAMTS9, which is actually not essential, but if it's knocked out in zebrafish, you get female infertility. So at the ADAMTS9 gene target, we're kind of playing with that combination of gene drives and sex ratio biasing. Currently, I am assembling genetic components and testing them in cell culture. So I'm not yet to the stage of injecting any of these components into the common carp. Gene drive resistance can occur when DNA is repaired by non-homologous end joining. So earlier I had mentioned that there are 2 different repair pathways that can happen when DNA is cut. The gene drive relies on Cas9 cutting the DNA. And in gene drives where you just cut it in one spot, If NHEJ puts it back together, then you can get a fish that can no longer be targeted by the gene drive, but will still survive. And then you can end up with a population that's resistant to your gene drive. In order to avoid this, we are using 2 guide RNAs instead of 1 to target the beginning and the end of the essential gene that we're targeting. So that if non-homologous end joining does put it back together, it'll result in a large deletion that won't allow the carp that inherits that to survive. And if it's repaired by HDR, then we get the expected result of the gene drive copying itself over into the wild type position that was cut. Guide RNA efficiency is currently being tested. I'm just expressing the Cas9 ubiquitously using the Carp beta actin promoter, and I'm testing 1 guide RNA at a time. I don't have results on this yet, unfortunately, but I will very soon. With the haplosufficiency, I kind of like drew out kind of like a larger picture of how it would work. So with an essential target that is haplosufficient, you release your gene drive carp, and at first you're mostly going to have the carp reproducing with wild type carp because they're just way outnumbered by wild type carp. And then you'll get the population being replaced by gene drive carp until eventually they start to mate with each other. And when they do that, they can only pass on 2 copies of the gene drive, and there isn't any copy of the wild type being passed on to keep them alive. So that's where the suppression happens. With sex ratio biasing, it would be a bit less aggressive in this case, or at least I think it would. We would probably do a bunch of modeling and testing to figure out for sure. But females that receive the gene drive will be infertile, and males will be able to survive and continue passing on the gene drive. So at first, again, it behaves very similarly. If a gene drive carp mates with a wild type carp, which is going to happen mostly when they're out there, you're going to get production of more gene drive carp. And eventually you'll have, again, those gene drive carp have to start mating with each other, spawning with each other, and then you'll produce adult females that are alive but sterile, as well as males that can continue to pass it on, whether they spawn with a gene drive female or a wild type. The onologues, though, are going to make this a lot more complicated. The haplosufficiency that I'm talking about, these genes having, was tested in zebrafish, which only have 2 copies. Some of the genes we're targeting in the carp, there are 4 copies. We have to figure out if those 4 copies are still functional. How many of those 4 copies are sufficient for it to survive? It might only need 1 functional copy to survive. It might need 3. We don't know that because it hasn't been tested in the carp. This diagram, the schematic here, and the other ones I've shown are assuming only 2 copies of a gene drive. There's also then the question of gene drive homing. You have that divergent evolution. When you have those extra copies, the sequence may have changed since that duplication happened. So the cutting efficiency of the guide RNA might vary depending on which of those onologues you're targeting. And also the ability of the gene drive to copy itself over or home into the wild type spot after it was cut might vary because, again, there will be that sequence difference. Which might make HDR less efficient or inefficient. So these are things that we are going to test in the common carp and see how it works. The hypothesis that we're going off of is that 1 copy of a gene that was haplosufficient in zebrafish will also be sufficient in carp, and that cutting efficiency will be equal for all of the functional copies as long as the guide RNA is designed within conserved regions, and that homing ability will be best for the onologue, like whichever one from which we took the homology and copied into our construct. But as long as if we can generate a carp that has it in all, like both onologues, then it should be able to pass along just fine into both. Even if we get good cutting at all of the sites, but homing is low, for one of the onologs, the NHEJ should still result in deletions in those other sites. So we shouldn't get resistance coming up from those onologs in the population. Oh, and we would test this by sequencing from genomic DNA of gene drive carp. And that AIM3 that I'm just going to kind of gloss over a little bit is Cas9 is that enzyme that causes the cutting. And there's literature showing that in cells as well as in vitro, that enzyme doesn't cut very well at low temperatures. Specifically, its activity significantly drops off at 25 degrees Celsius. This is a problem because the carp, and really anything that you're going to design genetic biocontrol using a Cas9 with, aren't at 25 degrees Celsius. I found 2009 global average temperatures by month, and I just kind of pulled out 3 of them at random to look at. And the problem here is the boxes where the average monthly temperature is high enough for Cas9 to be active are not very big. And they'll get even smaller when you consider that these are land temperatures. So this doesn't account for differences between, like, this doesn't count for water being colder than the land, and these are averages. So this doesn't account for daily fluctuations or nighttime, daytime fluctuations. So if an animal has a Cas9 that isn't active during a time because the temperature is too low, then the genetic biocontrol itself might not work. This could be a problem in the genetic biocontrol of invasive carp. We're using Cas9 nuclease for gene drives. We're using enzymatically dead Cas9 for some of our other constructs. In the lab where we have our carp, they're always between 15 and 25 degrees Celsius, which is below the temperature that Cas9 likes to work. I'm not going to touch on this too much, but we've also seen in some fruit fly tests using Cas9 that one of our genetic biocontrol methods was not effective once the temperature dropped down to 18 degrees as compared to 28 degrees. So that was kind of proof that this problem exists. So there are 3 different main ways that this could be happening from an enzyme perspective. It could just be that the enzyme doesn't cut well at the cold temperatures. It could be that the enzyme doesn't stick to things well at cold temperatures, or it could be that you just don't have good expression of the enzyme or RNA at those colder temperatures. Our main hypothesis is that it's the actual catalysis that has a problem, especially because in some tests using an enzymatically dead Cas9 activator, it was working fine at temperatures down to 4 degrees Celsius. So this is an enzyme that doesn't need to cut using the nuclease in order to work. It just needs to bind and get a thing fused to that enzymatically dead Cas9 close enough to the right location. We're doing this using directed evolution, which is a strategy where we create a whole bunch of mutations and then select for the ones that are, in this case, functional at cold temperatures, and then mutate them again and keep going in cycles until we eventually get one that's functional when we want it to be functional. There's kind of a flow through here of what I'm doing. Really, we're just putting a plasmid into yeast that has the mutated Cas9s, as well as an antibiotic resistance gene that is intentionally non-functional. If the Cas9 cuts, it'll restore resistance, and then we can pick colonies that have that restored antibiotic resistance and then use them for the next round of error-prone PCR. I'm currently doing positive and negative controls for this, and I don't have results yet. Finally, one of the big things that we're excited to do with this genetic biocontrol project is engaging with stakeholders, regulators, and managers to determine how we should progress through technology readiness levels. So first, we're adapting the NASA technology readiness levels to be able to communicate how the technology of genetic biocontrol in Carp, for example, are progressing along and what stage of development we're at. And we're also trying to determine, as a group, a very interdisciplinary group, what should we be doing from a regulation level or reaching out with stakeholders or tribal communities at these different technology development levels. How soon should we be engaging? So that's something that I've been a part of, and I'm really excited to be a part of that team. So the next steps are I'm going to finish validating the guide RNAs in cell culture. I'm going to do amplicon sequencing of cells that I just transfected with a Cas9 and a guide RNA and see which ones had cutting occurring. And then once I do that, I can pick the guide RNAs that I'll actually use for gene drives and finish assembling those constructs and inject those into common carp embryos. I also would like to micro-inject the gene drives into the common carp embryos themselves instead of just doing it to test the guide RNAs. And then once they mature, test for cutting efficiency and homing ability. There will be crossing to wild type carp and seeing what happens. And I'd also like to do a split gene drive, which I'm not going to dig into too much here, but the idea is you make it so that the gene drive kind of has a timeout system built into it. So it's less aggressive and easier to control. The idea is you have the Cas9 present somewhere in the genome, but it won't be carried along by the gene drive, only the guide RNAs will. So over time, you lose the Cas9 and it no longer works unless you reintroduce Cas9. For the directed evolution, I'd like to finish the positive and negative controls and actually begin the directed evolution to make my enzyme work in colder temperatures. And then these are some like future directions, but I'd like to investigate more types of gene drives, maybe find more ways to combine gene drives with sex ratio biasing, looking into gene drives that work with enzymes other than CRISPR-Cas9, maybe TALEN-based gene drives will work better at colder temperatures. I'd also like to do those directed evolution experiment with different Cas9 variants, maybe like C-base editors or those enzymatically dead Cas9 activators. Once I get Cas9 variants that work in the cold, I'll do a whole bunch of different things to evaluate them and test them. And then ideally, I would get to test how the cold-tolerant Cas9 nuclease compares with the wild type in actual genetic biocontrol in the common carp. I'd like to take a moment to acknowledge all of the amazing people in my lab that have helped me with my project, and also some collaborators and the funders that have helped. I would not be able to do any of this research without funding. So thanks for those that are funding the project. I'd also like to thank my committee who are listed below. So with that, hopefully I finished on time. Are there any questions?
We do have a question that is popped up. So this is looking at if there's any existing plans or anything to do gene studies at a lake or pond level.
We have actually done a little bit of looking at some local populations of carp. Someone else who used to be in my lab, his name is Nick, reached out and got some fin clips from common carp in lakes, and then looked at the DNA from those carp to see for one of the types of genetic biocontrol we were designing, how similar are the sequences of our lab carp to the populations in Minnesota. Does that answer the question, or was there something more specifically?
They're mentioning kind of a timeline. I just wonder if we're thinking along the lines of like when could we start using this? When can we start releasing
Oh, that's going to be quite a while. We
Yeah.
wouldn't want to just start releasing them out into the water until we're sure that it's effective and safe. Once we actually obtain genetic biocontrol carp, in my case, gene drives, but really with any of the ones that we're working on, we're going to first just test them in our lab. So we'll take carp. We have a tank of carp that were harvested from Long Lake, that we got in collaboration with Shemek. So thanks, Shemek, if you're watching. And we would first just cross them with carp and then observe what happens to their embryos. Assuming that goes well, we do pond trials where it's an artificial pond with bird netting, make sure that those carp absolutely can't leave, and then do like larger studies with wild carp. And then, assuming that we get all of the regulatory approval and we can do enough pond trials to show that it is effective, we would test it in small enclosed bodies of water following that. So
Thinking about it, I was wondering, I know zebrafish have been very heavily studied and some of the information you mentioned. Are zebrafish kind of the model that a lot of the basis for biocontrol, that genetic, is that kind of the baseline, and where
a
are they
lot of
at even this
genetic biocontrol research has actually been done in the fruit fly, Drosophila melanogaster, because it's really being primarily like focused on as a method to remove mosquitoes, because it's species specific and there are only certain species of mosquitoes that carry human diseases. Our lab is trying to focus on adapting those methods designed for insects to work in fish. Some of ours, specifically female lethal, and one I didn't touch on, EGI, engineered genetic incompatibility, we are testing at least components of them in the zebrafish because they mature a lot faster. The common carp takes 2 years to reach sexual maturity, so they're definitely not fast. But we aren't testing like the full genetic biocontrol in the zebrafish, and I don't know of anyone else that is either. So the question is kind of touching on like NHEJ is rare in C. elegans germline compared with in somatic cells. Is this also true in fish? In fish, I don't know about germline specifically, but I know that just in general, NHEJ is preferred. I personally have not found or really looked for the answer to that question, so I'm actually going to write that one down and look into that a bit. For the common carp specifically, I know that that data is not out there. I haven't found it, at least if it is. But for just fish in general, I don't know.
Well, thank you so much
Yeah,
for your time today. Your presentation was wonderful.
thank you.